Hi, good afternoon. Thank you for the introduction. Uh, so there are over 100 million compounds reported in databases such as PubChem. We have over 35 million publications in Medline. There is information about the genetic determinants of more than 30,000 different diseases. Nevertheless, the FDA approved only 37 drugs last year. So I think that we can confidently say that all of this wealth of knowledge that is available is not maybe being exploited into the drug discovery process. And what are some of the reasons for that? Well, some of the reasons, not all, might be related to these three challenges. First, that the relevant biomedical information that might be related to, for example, the diseases, it's not readily available in one single resource, but is scattered among many different repositories that you need to locate and understand. Secondly, because yes, structured data is cool and it's good, but the most recent findings are only reported in the literature, in the scientific literature, and it takes some time for this finding to being collated into expert-curated resources. And third, because well, once you located the information, you have to integrate it and put it together. So to solve these issues is why we have developed DISGENET Plus, that builds upon over a decade of experience of our group, like our academic group, then turned into a startup that is called Bioinformatics Solutions. And in this DISGENET Plus, we solve these issues, and we put together all the information available about the genetic determinants of human diseases. So what is DISGENET Plus? Well, it's a platform that first features a comprehensive knowledge base that standardizes and integrates information about genes and genomic variants associated to human diseases, but also has information about drugs. We cover all different therapeutic areas in our database, meaning that we have information for different disease classes, for cancer, but we also include information about human traits and drug adverse events. And finally, DISGENET Plus is an interoperable resource because we annotate all the information using standards and ontologies to make it easy for you to, for example, integrate it with your own data. So how do we develop the database? Well, we first, as I mentioned, integrate data from the most popular repositories that contain information about, for example, genomic variants associated to diseases or gene disease associations, but we also take information from other useful resources like human phenotype ontology, the disease ontology, to annotate, for example, the diseases, and information like from databases such as NOMAD to also put into context the information that we have, like, for example, what is the allelic frequency of the variants, the pathogenic variants that we have, or the disease-associated variant that we have in the database. And we also unify this information with data coming from the scientific literature. So as you know, over 100 scientific articles are published each year in the field of disease genomics. So to keep pace with this volume of new information, you need to use text mining approaches, natural language processing approaches. And for this, we have developed our own text mining tool that looks a little bit like this in a very in a simplification, let's say. So we start with the 35 million publications that we have in PubMed, and then we perform using a machine learning approach, we perform a document selection so as not to process all the information, and also to keep the information that is relevant for us related to disease genomics. And in the first step, after this, we perform a name entity recognition step using a dictionary-based approach. And our dictionary contains information about billions of entities, which are diseases, genes, variants, and also drugs. After this, we perform a relation extraction approach, and we end up having over 5 million relationships between genes and variants. So to illustrate a little bit how this looks like in the database, and to explain and to give more details about the process, I am showing 2 sentences that come from 2 different publications. And as in different colors, I am showing the different types of entity that we have in the database, like diseases, genes, variants, and drugs. And as I mentioned in the first step, we are able to not only detect that this entity has been mentioned, like this drug or this gene, but an important step is the normalization of the entity, which makes it easy then to analyze this data afterwards and to put it in the context with the curated expert data that we extract from curated databases, right? So after this normalization step, in which we use, for example, concept unit identifiers for the diseases or an NCBI gene identifiers or DBSNPs for the genes, we perform a relation extraction in which we improve this step of entity recognition, and we filter and we keep only sentences and relationships in which we have a gene that is mentioned in the same context that a disease. So after doing all this process and putting together all the information from the scientific literature and from the curated databases, we have a lot of information, right? So there is information for over 25,000 genes. Almost every protein coding gene is present in our database, and for 600,000 variants. And we have information for 36,000 diseases and phenotypes, and for 3,000 drugs. And we have information we have millions of relationships between the different entities that we have in the database. So that is a large volume of information. How do we navigate it, right? But for that, we have our own metrics that we have designed. First of all, the DISGENET Plus score that takes into account not only the number of databases that report an association, but also the number of publications. So to give you more or less an idea of how this looks like, the DISGENET score tends to favor a little bit or a lot associations that are from the Mendelian kind. Like, for example, Mendelian diseases in which the gene is caused, there is one single gene that has mutations that cause the disease. As you can see, these associations are here on the left, and they have a score of 1, while other very well-established associations related mostly to complex diseases that you can see on the right, they have a lot of publications, but they still have a 0.5 score because they are not reported by every single database. And it makes sense because the original databases are typically sometimes focused on one type of disease, like, for example, Mendelian diseases, right? So this means that if you have a high score, it's a very established and very well-described association. And if you have a lower score, then it might be it's also an interesting association that you can combine with other information that we have in the database to say, oh, this is novel, this can also be interesting, right? Other things that we have, other metrics that we have that can be useful to the user to filter the information and to rank is the evidence index. So we also have artificial intelligence approaches to detect negations in the sentence. And then we quantify the number of let's say, positive associations with respect to the total. So to design what we call evidence index, which is closer to 1 when it's supposed to every sentence, every statement in the database is saying, yes, this gene or this variant is associated to this disease. And if it's lower than 1, it means that there is some contradictory evidences. And this doesn't mean that this is wrong, because, for example, in the context of GWAS, some results have not been able to be reproduced, right? So in some population, this variant will not be associated to this disease. And it's also like a correct result. And then you can use this also to filter the information that you have in DISGENET Plus. We also classify the relationships between the genes and the diseases and the variants of the diseases using different, what we call the DISGENET Plus ontology that looks a little bit like this. And I want to illustrate just with a couple of examples. Like it allows you to refine the information about the gene disease association. Like you can find genes which is expression have been altered in the context of a disease, or, for example, genes that suffered a post-translational modification in the context of the disease. So you can actually prioritize what you are most interested in, maybe. And finally, we have other metrics and ontologies, as I mentioned before, to annotate genes and variants. And for example, the disease specificity index that allows you to find genes that are more specific to the disease, or more pleiotropic genes that are perturbed, like TNF and other interleukins that are being altered in many diseases. So we have a lot of data, we know how to navigate it, and now what do we do with it? So we envision like 3 typically use cases or scenarios, right? So first, the drug and research and development, in which you, for example, might be interested in developing a new drug for a disease, and then you can find what are the targets of the genes associated into this disease in DISGENET Plus, and you can identify some interesting candidates. Second, given that we have adverse effects also in the platform, you can check if your target that you're studying is associated to some of these adverse events in DISGENET Plus. And you can use it also to find novel indications, like it's a gene that is already the target, it's probably associated to other diseases. Can I use it also for this other disease? Another scenario would be in clinical genomics. Typically, you can have a sequencing experiment, the result of many variants, and maybe you wonder, has my variant or the variant of my patient been found before, or associated to this phenotype, to this disease, or even to a similar disease, that you can also use DISGENET Plus to find out similar diseases. And finally, and more exciting, I think, is for the development of AI applications for different things. Like typically, DISGENET has been used a lot for designing, for example, for training machine learning systems that allow you to predict, for example, gene disease associations, right? And more recently, it has been used also a lot to include into the knowledge graph as a source of gene disease and variant disease associations. In order to further illustrate what you can do with DISGENET, then I wanted to showcase an example, because I am not going to argue that the genetics is important in drug development, because many people, the people that were here this morning, you could hear that it's important, and there have been several papers lately that say that yes, when you have a genetic support, this leads to improved therapies. And actually, in the morning, there was a paper that discussed that a fraction of the 2/3 of the drug approvals in 2021 were supported by genetic evidence. And then we wanted to, we wondered, then can we do better maybe than 2/3? And this is the result. So I'm guessing that the people in the back cannot see anything, but what I'm showing is in the y-axis the drugs that were approved in the 2021, and in the x-axis you have the diseases, the indications for these drugs, no? And in the heat map, what I'm showing is the gene, the target gene of the drug. And we are coloring the gene by a score. So if it's really blue, it has a high score. And you cannot see, but I'm telling you that some of the genes have a very high score, because the drug has been designed to target the gene that causes the disease, like, for example, Pompe disease, or there is also for Alzheimer's disease, APP, which is one of the most reported genes associated to Alzheimer's disease. But for other, we have a variety of scores, so you have different strengths of the association, let's say. And in summary, we found that if you do, because of the more rich representation of phenotypes that we have in the database, because we use another vocabulary, which is UMLS, we are able to better map the indications for the drug. And we obtained that we have 90% of the approvals in 2021 are supported by genetic evidence, and which surface, it's a little bit larger than the publicly available resource. If you want to know more details, like a lot of details about the analysis, you have the link there, so you can visit there and see how we did the mappings, what information do we have. To conclude, DISGENET Plus, it's a repository containing information on disease genomics that stores the knowledge, the set that has been stored in 35 million publications. It also contains information about different types of, for example, biomarkers, and it has metrics and attributes to rank the information. And I think we think that is a versatile tool that can use for many applications. If you want to try it for yourself, you can register for a free trial at disgenetplus.com. And if you want more information, you can also contact us. Thank you very much for the attention.
I will make a first question, even if it's not a privilege. Then I go to you. So I have a doubt concerning your last example of the 90%, because this is only the drug that has been approved last year.
I think it's 2021. 2021.
Okay, but it's 1 year. The 66 is for the whole.
No, the 66 is for 2021. 2021.
Okay.
It's not even last one. Because I'm playing with this, the 219,
Okay,
220,
could be
221, and
correct. It was-
222, and it's more or less
could be-
the same result.
Because the 50 for all the ones that were before 2015, the 66, I don't know. So we had one question there.
Thank you. So, if I'm understanding correctly, are you essentially using a bag of words approach after doing the named entity recognition, and then using co-occurrence as a proxy for association? No.
No, and this is important because many available resources do
Yeah.
only co-occurrences, and I'm sorry, but the data, our data is cleaner, let's say. We are actually using different, so we do the name entity recognition approach, and then we find the entity, and then we have a classifier for genes and for diseases, just for genes and for diseases. So we get a threshold that of a machine learning system that, or a model, these language models that were pre-trained that we fine-tuned, and they tell me this is a 70% chance of this being a gene, there is an 80% chance of this being a disease. So we filter out things, right? And then we have another machine learning model that tells me this is a relationship between this gene and this disease.
Right.
And then we have another machine learning model that tells me this is an altered expression, this is a post-translational modification, this is genetic variation. So.
Yeah, that's super important because you could have something in the literature that says this gene is not associated with this, and if you do bag of words approach, it would not.
It would say yes. But still, you can capture this. In fact, you capture this because it's not association; it's just a relationship.
Sure. Yeah.
What it is not a relationship is when you have an abstract and you have Duchenne muscular dystrophy. A parenthesis, DMD, it's a disease that causes this and that. And then you say, DMD, this is a gene, and this is a disease, right? And unless you like, I have some examples of the thing that can happen if you do not do relation extraction, like properly relation.
I suggest, and then you can keep discussing. If not, we don't have the time for the other.
Thank you for your talk. I had a quick question whether the tool is free, but you answered it on your last slide. But a follow-up question is, I think a big important part about gathering information is for machine learning models, especially for these kind of works, is how do you visualize and present that information? Because I've seen similar tools that also exist, and I've actually played with one. So I was just wondering if you can comment on your efforts to make this information more easily accessible to the users.
I can only tell you that you can register at the website, and then you will see how it's summarization of the information is very important. So this is some things that we do. So we present different metrics and summarization, like to find rapidly how many publications are associated to this gene, to this disease, to this gene disease association, like what is the association type of, what is the impact of this variant, what is the allelic frequency. So you have in the web interface, you have a lot of way of playing and filtering and ranking, combining filters to just keep the information that you are interested in. And furthermore, we also have visualizations.
All right, last question.
Hi, this is also related to this plot. So some of the scores are middle ranking, which is fair. But the question is, would these be the top ranking genes if I ran the association? If I look
No,
for association?
not for all. Not for all. Yes, that's a good question.